You have reached the critical final 2 weeks of your outdoor or greenhouse growing season. At this exact moment, many cultivators execute a heavy intervention. They begin aggressively flooding their large container soils and living beds with pure, pH-balanced water. The intent is to starve the root zone, force a sudden nutrient deficit, and compel the plant to purge its internal chemical reserves before harvest. In practice, this heavy saturation routinely causes catastrophic side effects. Flooding the substrate degrades the soil structure and creates root zone hypoxia. The excess moisture also severely spikes the ambient humidity in the greenhouse, providing the perfect breeding ground for a crop-destroying outbreak of Botrytis cinerea, commonly known as bud rot. Yet cultivators have risked this practice for decades, driven by the belief that forcing the plant into starvation is the only way to prevent acrid smoke and dark ash. Controlled scientific trials have disproven this belief. Complete nutrient deprivation does absolutely nothing to deplete the actual mineral composition stored inside the flower. If starvation fails to clear these minerals, the drivers of crop quality must reside in a different biological process. We have to look at what actually happens inside the soil and the plant stems when nutrition is abruptly cut off. To understand why reverse leaching minerals out of a living plant is a physical impossibility, we need to zoom in on the microscopic mechanics of the plant-soil interface and trace the exact journey of a water molecule. This diagram shows a cross-section of a plant root tip surrounded by soil. When pure water hits the substrate, soluble salts are quickly leached away. This aggressive wash causes the osmotic gradient of the soil solution to shift, lowering the electrical conductivity outside the plant. But the system hits a physiological wall. Root-applied water cannot create a reverse vacuum. The water flows inward, but it cannot pull integrated nutrients back out of the root system. Once inorganic ions cross the root's endodermis barrier and move into the plant, their state changes. They become permanently locked into organic complexes, woven into cellular walls, and packed into vacuolar storage compartments. This one-way biological transport system means that pure water in the soil is mechanically incapable of cleansing floral tissue above the ground. The physical limits of the endodermis render root zone flushing a flawed strategy for purging minerals. This presents a glaring contradiction for cultivators. If minerals are not leaving the flowers, why do a plant's large fan leaves visibly turn yellow and die off during a heavy flush? You are looking at a desperate survival mechanism. When the root zone runs out of food, the plant triages its resources. Immobile nutrients like calcium, iron, and sulfur remain fixed exactly where they are. To keep the active reproductive sites alive, the plant starts cannibalizing its older vegetative leaves to relocate mobile nutrients, particularly nitrogen. This physiological response is called programmed senescence. The plant breaks down its leaf proteins, causing the localized yellowing you see radiating outward. It sacrifices its solar panels to protect the flowers, rather than executing a systemic mineral purge. Please like and subscribe, it really helps our channel a lot. That classic yellow fade signals that the plant is actively digesting its own vegetative tissue to survive acute root zone starvation. Knowing that heavy flushing causes stress without removing chemicals, the scientifically validated alternative is a targeted late-stage biological taper. For synthetic greenhouse container soils, cultivators implement a subtle 5 to 7 day nutrient taper. By reducing fertilizer strength by exactly 50%, the plant utilizes the residual nutrients in the soil while avoiding the root hypoxia that comes from aggressive water saturation. Living organic soil beds require a stricter protocol. Cultivators must avoid pure water flushing. Instead, they suspend liquid nutrient inputs and maintain soil moisture based strictly on the plant's evapotranspiration demand, replacing only the water the plant actively uses. By matching the taper strategy to the physical structure of the soil, growers preserve the delicate biological food web. They also achieve the same operational input cost savings that first made traditional flushing so popular, but without damaging the crop. With the roots protected in the greenhouse, we have to look to the drying room to engineer the actual smoke quality. The smooth smoke and white ash cultivators desire are driven by post-harvest enzymatic catabolism. This is the biological process where specific enzymes naturally break down bitter chlorophyll molecules and complex starches in the dying tissue. That enzymatic reaction requires precise environmental triggers to function. To break down those harsh compounds, the drying rooms must be strictly maintained at 15 to 18 degrees Celsius, or 59 to 64 degrees Fahrenheit. This precision gauge illustrates the final critical requirement: water activity. The final product's moisture threshold must be targeted strictly between 0.55 and 0.62. Anything outside this narrow window stalls the enzymatic breakdown and destroys fragile volatile sulfur compounds, regardless of how the plant was grown. Thanks for watching. This content does not constitute medical advice, legal advice, or recommendations for the use, self-treatment, or consumption of any substance.